Hi, this is Remembering the Past, the show where we talk about people who died recently who've had a profound effect on our history, our society, or our culture. And in our ongoing series on Nobel Prize winners, tonight we're going to talk about Irwin Rose, who died recently at the age of 88. And Irwin Rose was one of the winners of the 2004 Nobel Prize in Chemistry. Dr. Rose was working at the Fox Chase Institute for Cancer Research in the 1970s when he was approached by two Israeli researchers, Avram Hershko and his graduate student Aaron Ciechanover. The three of them set about to solve the problem of how a cell gets rid of proteins that the cell no longer needs for functioning. When certain proteins in a cell are no longer useful to the cell, the protein has to be destroyed by the cell and its component parts can be reused. No one knew how that was done, and the Israeli researchers and Dr. Rose settled on a protein that was found in cells that no one knew the function of called ubiquitin. They called it that because it was found all over the inside of the cell, and as I said, no one knew what its function was. Rose and the other researchers figured out that it was essentially the assassin. That is, ubiquitin would target proteins that the cell no longer needed and would destroy them. It would bind to them, that's where the chemistry came in, and it was called the kiss of death, because once the ubiquitin bound to those proteins, they would be destroyed. It was this research that led the three of them to share the Nobel Prize. Here's Nobel.org with comments from Dr. Rose and Dr. Hershko on how a cell destroys proteins that it no longer uses.
Within the human body, proteins carry out a range of functions essential to life. This year's Nobel Prize in Chemistry has been awarded for the mechanism of protein degradation in cells. It was known that proteins in cells are degraded when they are not needed anymore, but no one knew how it was done. Having identified ubiquitin, a small protein isolated in 1975 as playing a key role in protein degradation, Hershko and Ciechanover set about trying to define the mechanism of degradation. This discovery came about during a series of sabbaticals at Fox Chase Cancer Center in Philadelphia, where they worked with the American scientist Irwin Rose.
Hershko, at that time, with his graduate student, Ciechanover, came to our lab in summers and occasionally for whole years, and we and they elaborated the different steps.
We found that the small protein, which we discovered here, its role is to tag the proteins for degradation. It is a kind of a death tag, or a kiss of death, it was called by the Swedish Academy. It's really a kiss of death for the protein, but I should say it is the kiss of life for the cell, because it's very important to degrade the right protein at the right time, otherwise the cell gets out of control.
So it's a small molecule ubiquitin that decides which protein will be executed, will terminate its life at a certain point.
The laureates worked out that proteins to be degraded in cells are tagged or labeled with a ubiquitin molecule. The labeled proteins are fed into structures within the cell called proteasomes, the cell's waste disposal units. It's here that they are cut into small fragments and destroyed. The medical implications of this discovery are far-reaching because proteins play a key role in cancer.
If you're going to understand the origin of the cancer, you've got to understand the control of a protein that influences the DNA.
Ubiquitin-mediated protein degradation, which is the system that we discovered, seems to be involved in almost every basic process in life. And that is because proteins are involved in almost every basic process in life. And that includes control of cell division, which is very important because when cells get out of control, then we have cancer. So this prize is a chemistry prize because it was done for understanding how the chemical processes are in the body. But actually, the big implications are for medicine.
This contribution to chemistry and medicine is the result of a remarkable team effort.
We are all a team. I know them very well. Aaron Ciechanover was my graduate student for 5 years.
Unlike Hershko and Ciechanover, Irwin Rose has never specialized in ubiquitin research.
It was not my whole life. So therefore, since it wasn't my whole life, I didn't feel that I was really that instrumental in it.
I am very, very glad that Irwin Rose was included in the most prestigious prize of all. And he deserves it because he had a very important impact at the most crucial period of this research.
Here's the actual awarding of the Nobel Prize to the two Israeli scientists and Dr. Rose in December 2004 in Stockholm.
Professor Ciechanover Professor Hershko, Professor Rose. Your discovery of a system for controlled protein degradation in cells has fundamentally changed our way of thinking about protein degradation. We can now understand at molecular level how the cell controls a number of central biochemical processes. More reactions regulated by ubiquitin-mediated protein degradation are identified every year. On behalf of the Royal Academy of Sciences, I wish to convey to you our warmest congratulations, and I now ask you to step forward to receive the Nobel Prize in Chemistry from the hands of His Majesty the King.
And the third laureate in chemistry is Dr. Irwin Rose.
Well, Irwin Rose was a nice Jewish boy born in Brooklyn in 1926. Notice how many of our Nobel Prize winners have that same history, born around the same place and about the same time. He moved out to the West Coast, and here he talks about his early educational background and what got him into biochemistry.
I have an anomalous story. There's no precedent for this. We moved from the East Coast to the town of Spokane, Washington when I was about 13 years old. I did not adapt very well to the style of the place, and I spent most of my time in the public library. And I enjoyed the company of the Journal of Biological Chemistry because it was the book-shaped thing. And in those days, it was the small journal. I was very unpopular with the other students. And so I read the small Journal of Biological Chemistry, and I found an article that I thought I understood. And I read it, and I thought I understood it to the point where I could make some suggestions about how it would be, the experiment might work. And then I was very satisfied with that. And then I didn't spend much time in science at that point. Went into the Navy, got out of the Navy, tried to go to University of California at Berkeley, but due to the failure to find a bulletin board announcing the laboratory time of organic chemistry, I couldn't do my organic chemistry there, so I went back to the State College of Washington. And there I was influenced, I would say, by the embryology teacher, who was a very strong personality in terms of academic research. He tried to encourage his students. I went to the University of Chicago, and it was a big shock to learn all the new kinds of things that they were teaching there in organic chemistry and that sort of stuff, and very attractive concepts. Things began to come together in my mind as to how chemistry worked and how I might be able to exploit some of the early kinds of techniques that were being used in organic chemistry into biochemistry, which is something I was attracted to due to my reading of the Journal of Biological Chemistry. At that point, I signed up. There was a big gymnasium, and people were signing people up for which major you were going to go into. So I said, okay, I'll be a biochemist. So I entered into the Department of Biochemistry. Never saw the chairman of biochemistry because he was the appointed ambassador to Britain for the United States. I floated around in the Department of Biochemistry and learned some interesting things, and then I never wanted to work with a mentor because I always wanted to have my own reputation and be free to do what I wanted to do. So I worked with the weakest people in the department. Don't make that public. That way I became to learn some more independence, and once in a while I did a good experiment, so I had more confidence that I could do research, and so that's how it got started.
Here Dr. Hershko talks about his first meeting with Dr. Rose.
So then I met Ernie at a meeting in 1976, one year before my sabbatical was due. And I remember we met in the breakfast. So I said, "Can I just begin to talk?"
It's all right. I forgot it.
Yeah. Breakfast table. So I knew he's very well known for his work on enzyme mechanism. That I knew. But then I asked him, "What are you interested in other things?" So it turned out that he was interested in protein degradation, and that was a secret. It was a secret because he never published anything on it. And I asked him, how come you never published anything on it? So he said, there is nothing worth publishing on protein degradation. And anyhow, so I liked that attitude very much, and I asked him, can I spend my sabbatical with you? And he said, yes. So that's how it started. And then Aaron, at the same year, he started his PhD with me. And after my sabbatical, the summer after my sabbatical, Aaron joined us, and then he joined us for a couple of summers afterwards. So that's how that's how the whole connection started.
Dr. Ciechanover talks about the skepticism they met from the scientific community.
And after this presentation of the professor, this was already 81 when we had like 8 or 9 papers already in the literature with huge amount of information there. And he was a protein researcher, and he raised his hand, I remember very well, and the other guy remembered also very well the incident. And he said, you know, I have a question to ask you. There is a fellow in Haifa by the name of Hershko, and another one with a very complicated Polish name that I cannot even pronounce, that published a series of papers on a small protein that is attached to other protein and marks them for degradation. Can you comment on it? And he basically dismissed it as an artifact. And I don't criticize him. All I'm telling you that it was symbolic for me that after 8 papers in the literature, this was the spirit in the field from people who worked in the field, and there were very few. As a matter of fact, it acts to our benefit. Because they left us alone for 7 successive years, even after I left the lab, to work out basically the entire system. The next scientist to join the field was a scientist at MIT, Alex Warshawski, that joined in 84 or 83, basically. We published in 84, and even I was there and we collaborated. So for 7 successive years, they let us lay the entire story down in the literature. So I don't criticize them. Actually, I appreciate them tremendously for letting us doing it in retrospect.
Dr. Rose reflects on that skepticism.
They're making observations, and the observations get published. So the observations are true. Whether anybody will say that belongs to a big story like it turns out to be, it's not predictable. But so you don't make claims like that. You say that this is very interesting, and so on and so on and so on, and you keep following it up. And it doesn't necessarily become the center of attention yet until you build a big enough story. I think that's the way it works. You have to add successively to the picture you're trying to portray. It's not sufficient to just provide data. So I think that's part of it. But I agree that it's important to be left alone for a sufficient amount of time in order to be able to do it and not feel that you're in the middle of a big activity already.
Dr. Rose talks about what he did at the end of his career.
When I retired from Fox Chase, I took my spectrophotometer and a lot of my chemicals, based on sort of the suggestion of his recommendation. So I took all my chemicals and my spectrophotometer and my constant temperature bath and so forth with me to Irvine. When the person whose laboratory I was sitting in decided to retire, I had to do something with the spectrophotometer, and so I found a place in my kitchen for it. And this was very convenient because it saved me a lot of time. I didn't have to go to work every day, and if I had a little experiment to do, I could do it in my kitchen. So that was very good. Although I've got a lot of chemicals that I have no use for. But I have a freezer, and I can make ice cubes, and I can break them up.
And here are Dr. Rose's final reflections.
I don't have any hobbies. You know, I'm very embarrassed when people ask me what are my hobbies. I don't have any hobbies. I mean, it's just enough to keep up with the things I'm trying to solve. You know, you work on little puzzles, so on and so forth. Each puzzle requires attention. You get an idea. You get your ideas at different times. Sometimes your wife makes a statement, and you say, aha, maybe you're right. And so you go off to your kitchen and do a little experiment. So that's the way you make progress if you continue these things. So that's my recommendation. Do not retire.
You hear what he said? Aha! Well, I'm going to close on that note. I want to thank my producer and IT genius, Sid Tepps. And since we're talking about Dr. Rose from Brooklyn, I thought we'd close with a song by a nice Jewish girl from Brooklyn, Barbara Streisand. She's singing about a rose from 2nd Avenue, which is on the Manhattan side of the Brooklyn Bridge, but I'm sure Dr. Rose had relatives on the Lower East Side, and he probably ventured over there occasionally also. So here's Dr. Rose landsman Barbara Streisand with a little bit of second-hand rose from Funny Girl.